Okay. So let me start out. Too bad I cannot see you guys, but let me start out by the looking at the syllabus. We can set the common ground so that the prerequisites is computing 2 and probability and statistics. And as first of all, I'm not going to take up the full class period today because on the Zoom it's not the same as doing in the classroom. So I'll just go over a few things for about 2 hours and then let you take a look at the homework and so forth. Okay. So you have a textbook that is reasonably priced. So you can, I expect you to have the textbook. And there are some online sources. And we're going to look at the bioinformatics in 2 parts. Over the last 20 years, particularly in the last 10, 15 years, the bioinformatics has grown so fast, so quickly, and that has to do a lot with computer programming. Okay, so with the computer programming, they are able to locate the different DNA sequences and gene sequences and things of that nature. So that computer algorithm, we're going to take a look in the first half. And the second half will be looking at some of the analysis or modeling techniques that are used when dealing with a large amount of data. So what they call a big data. The kind of work that you're going to do will not be a really big data, but at least have a sense of what kind of data modeling or data analysis can be done given a reasonable amount of data. Okay, so that's what we're going to look. I cannot see it, right? Okay. So if you have the machine learning class already, or you're taking machine learning class, then you'll be in a huge advantage because the kind of data modeling you're going to do will be a subset of machine learning. Right? So I don't know how many of you are either taking it or taken the machine learning, but that's what you have. So here are some of the algorithm part. And then in between, I'll fit in the data models so that we can look at how to analyze the data. Some related to the bio data. And for the assignments, you have basically a weekly assignment. And when you have a programming assignment, I expect you to do in Python language. If you don't know how the Python language works, then it's a fairly simple language, so you can pick it up in a week or 2. So the homework 1 today will not involve programming, and next week you have a very simple programming to do, and with that, I'm sure that you can pick up Python language right away. And evaluation is listed there, and you have a weekly quiz, and you are sort of excused from 2 quizzes. In other words, you can miss 2 classes. and midterm test and final test or project. And program assignments or homework assignments will count for 20%. And with the ChatGPT and so forth, I cannot put too much weight on the program assignment nowadays. Okay, that's the fact of life. Okay. Okay, any question on the syllabus or overall organization of the course? We can put the comments in the chat. Say 22 is when you have a half the class. Okay. So are you all good so far? Okay, let's then move on to the slides that you also have in the Blackboard. Put it on. Okay. And start off with the first slide. Share that. Okay, so bioinformatics is a fairly new field, and it started from probably 15 years ago or so, when people realized, particularly in the biology, that with the more powerful the computers and so forth, they can do a lot of computations or the algorithmic execution with the computers when they do the sequence search and genomic comparisons and protein modeling and so forth. Okay? So you may be wondering what is involved in bioinformatics. So actually, the basically three fields all combined. Biology, obviously, and mathematics, particularly probability statistics and the linear algebra. And I'm going to assume that most of you do not have a linear algebra in the background. So I'm going to cover that later on for the portion that we really need. But if you have a linear algebra already, you'll be in a huge advantage, just like you had a machine learning class. And obviously also the computer science, because you have to develop algorithms and run them to come up with some kind of outputs and outcomes related to the biology, particular problem in the biology. And machine learning is really the data modeling techniques, and there are a few of them, and we'll cover the basics of them later in the course. So these are a few the kind of preambles, and it turns out that okay, the human genome was or the decoded the over 10 years ago, 20 years ago, and it turns out that practically all the genomes in individuals are identical except for 0.1%. Right? So it's a very the tiny portion of the genome that makes up the differences among us. And if you think about it, it's really the amazing how such a small difference can make every individual so different. So the one obvious question that we can ask is, if the huge majority of or most of the genomes are identical, then what makes us different with the different eye colors and different height and or things of that nature? And if anybody have an idea how come 0.1% of a genomic difference can make us so different? So if I pose the question that only the less than 1% of genomes are different amongst us, how can they be all we can be all different? It turns out that there are a lot more involved than just the differences in the genomic sequences. There are it has more to do with which genes are expressed or being active than what genes are there in the genomic sequence. So we'll get to that later. And also a significant question has to do with the next one. So if each cell has, we all know that each cell has the same set of the chromosomes and same set of the genomic sequences, then how does one cell becomes a skin cell and another cell becomes a hair and so forth? So how does a cell can tell how it has to be developed depending on where it is located? So that's a sort of the fundamental question. And the same with the stem cell. So if you have a stem cell, if you implant a stem cell, for example, in a liver, then how does it know that it is developed into a liver cell, not the skin cell or other cells? Yeah. So that's the another big question that you can ask. And also the bioinformatics are primarily used to study mutations in genomes, and mutations in genes trigger some of the diseases to develop. And from that identification of which genes are involved for which disease, they can use a number of different techniques to study the drugs development. For a drug development, it usually takes about 10 years and $1 billion or more, because you have to go through the animal testing and human test and so forth. So these are sort of more of biological questions. And the third one, the drug development, is really what triggered the fostered or the biotech companies in and around Boston. But we are not, obviously, we are not getting into drug development or biologically oriented questions, but we are looking at the computer science oriented questions such as algorithmic development. So the actual the algorithm that we're going to take a look at is to first compare the sort of the scan the genomic sequence to find out how frequent a particular sequence appears, things of that nature, and also how to finding the sequences with the similar subsets that are detected in a group of sequences and things of that nature. So we're more geared toward the algorithmic development than biology-oriented questions. So what we're going to do today is look at so this is a biology primer for CS majors. For some of you, this may be just a recap, and some just the recalling what you learned earlier or reviewing what you already know. and consists of two parts. One is DNA, RNA, and this part I'm going to go real quick. And second part is the protein, the protein manufacturing. So how the genes, how the proteins are manufactured from particular gene sequence. And the basic overall, the underlying the doctrine or theorem or theory is that it goes through two steps. One is the transcription followed by translation, and that's what we're going to look at in the second part of today. So DNA, RNA. So DNA, we all know that consists of a sequence of what's called the bases or nucleotides. And we have only 4 nucleotides in the ACGT, adenine, cytosine, and so forth, right? AGCT, and you have the models for each base. And DNA and RNA are basically the same, except for what substrate they are based on, and how the slightly different in the chemical composition of the two. So we're going to look at CTAG 4 nucleotides, and they are grouped into 2. Pyrimidine is C and T. And when in DNA sequence, it is a T, but if the same base is found in the yeah, I think there is, yeah. Same base is found in the RNA, then you have a slight difference in the chemical composition, and it's called a U. So T and U are basically the same. As soon as the nucleotide T in DNA is transitions over to RNA, then it changes form to U. So the pyridamine has a single ring, as shown in the model, and purine A and G both have these double rings. So they are, A and G are somewhat heavier than C and T. And because of this, the combination will be slightly different, as we'll see that later on. So you have a AGCT, and the AG is a purine group. So if you're not sure which one it is, whether it's an A or G, then they sometimes group it and call it R for a purine group and pyridamine group. So if you don't distinguish, excuse me, excuse me, T and C in the pyridamine, then you can group them and call them Y. And sometimes you need to group A and T together. And if you do, it's called a W and S for GC and different type, right? Different grouping you can have. And if you don't know which one it is, or group all of 4 bases together, then it's called the N. So for the non-distinct or something like that. So they have different letters to indicate which group of bases you are referring to. And DNA and RNA both are running across the substrate or the base. And depending on which base is attached, it is going to call the TG, this is C, yeah, TGCA. Okay. And there is a slight difference in the base between RNA and DNA, but I don't think we need to get into that. Yeah, DNA, we all know, has the double strand. So the two strands are sort of going parallel, but in helical fashion. And they are tied together between two bases from two different strands. Yeah. And this structure was first discovered by Watson and Crick in 1953, and they both got Nobel Prizes for their discovery. Yeah. And if you look at the dimension of the double helix structure, then the outer diameter, so the outer structure is about 20 angstrom. Angstrom is the, what is it? The 0.1 nanometer, basically. So 10 to the power of negative 10 meter, so 0.1 nanometer. And if you take from one place to the next one around a complete turn, then it usually contains about 10 base pairs, 10 bases. Yeah. And that the length is about 34 angstrom. So each base takes up, as you saw, the shapes of each base is a little bit different. And the double ring and the single ring and so forth, but each base can be averaged out to about 3 angstrom, a little bit over 3 angstrom size. So you have 10 bases for 3.4 nanometers, or basically 34 angstrom. I don't know if I have a symbol for that. Yeah, I don't have it. Okay, so they put usually circle on top of letter A. So that's the symbol for angstrom. We have an angstrom there. In human genome, we have 3 billion bases in the entire genomic sequence. And obviously they are divided into 24, 48 the chromosomes. So each chromosome will be much smaller. But altogether, if you stretch them out next to each other, or 24 chromosomes, then you'll have 3 billion bases. So if you line them up next to each other in a long linear sequence, then you have for 10 bases, you have a 3.4 nanometers, but we have a 3 billion, or 3 times 10 to the power of 9 bases of them. And if you do the computation, that it comes out to be about, so these two cancel out, and we have a 9 over 10, so approximately 1 meter. And 1 meter is about a yard, which is about 3 feet. So it's a fairly long sequence if you put them all next to each other. So the question is, but we know how small, tiny each individual cells are in our bodies. Our bodies also have about 3 billion cells. And I think over the period of 7 years, every cell is replaced by a new cell. Some faster than others, but so if you know you have a tiny cells, and chromosomes and genomic sequence are stored inside those tiny cells, how can this 3 feet long, the genomic sequence, so again, that's cut down into the chromosomes, how can they be stored into those tiny cells? And that has to do with the chromatin, or the chromatin structure. So when you have a long DNA sequence or some chromosome sequence, then they are not just put in a linear form. They are tied around this little knots, little beads, and then connected to the next bead. And sometimes these beads are connected next to each other to form much tighter structure. And these beads are the each of these beads consists of 4 histones, the other proteins, histone groups. So H1 and S4. And if you look at each bead, you have these 4, the histone group, and you have a pair of them, top and bottom. And DNA strand is kind of circling around, making 2 rounds, and then they connect it to the next bead. And then histone H1 works as sort of the closer. So it closes this particular DNA sequence that's connected to or tied to this histone, this particular histone, the core or histone, the bead. Okay. So that's how they reduce the size of the chromosome so that they can fit in the tiny, tiny little cell. We'll take a look at it later. Okay. So next, the well-known property of DNA sequence is that they have a double helix structure, and then between 2 helical structures, you have 2 bases that are connected. Okay. And when they're connected, they're always connected between pairs of AT or CG. In other words, A always pairs with T, the vice versa, T always pairs with A, and C always pairs with G. So there's no other cross connection at all. You can take a look at the YouTube. So this one, I don't think, yeah, so that's the property. And these bases are going to be, the nucleotides will be connected by hydrogen bonds, not only, yeah. So you have 2 bases, say A and T, they are connected by hydrogen bonds. And if you take a look at the hydrogen bonds between C and G, since it's connected to the C and G, they have 3 hydrogen bonds, 3 bonds between C and G. And A and T, or A and U, you only have 2 bonds. And this has the significance that it's much easier to break AT bonding than CG bonding, because you have 3 of them in BCG. So when you can kind of imagine that this one is running in one strand, this one is running in the second strand, the same over here, and they are tied to each other by these hydrogen bonds. So it's much easier to break the double strands if you have an AT binding between the strands. So then it's much easier to break them apart than CG binding. Now, prior to the Watson and Crick's discovery of the double helical structure, the orange Shargov studied the DNA structure extensively using the X-ray, the pictures, X-ray shots, and then counting which atoms are where and things of that nature. And prior to Watson and Crick, Orin Shargov has already observed or counted the number of the nucleotides A and T's in DNA sequences, and he already established the relationship. Okay. Okay, so this is let me sharing. Let me share the desktop now. See, I need to, yeah. So you have double strands running across, say, conceptually drawn this way. And we all know that A always pairs with T, T and A, CG, and GC, right? So if you count the total number of A and T's in the double strands, you can easily see the number of A's should be exactly same as number of T's, right? T's. Since you have, if you have a X number of A's, then you have to have a corresponding number of T's. And the same with the number of C and number of G's. So you can easily see that they have to be the same. And he actually verified them by actual physical counting of the nucleotides A and T's and C and G's already in 1951, but he couldn't put together into that the helix form structure. And he then asked, so this is called the Oren-Shargov's first parity rule. He then asked, in a single strand, in a double strand is obvious, right? Double strand is obvious, but in a single strand, would there be some relationship between A and T's? T, C, G. What do you think? What if the relationship, if I know the number of A's in one strand, would they be similar, close to number of T's in the same strand, same single strand? There's no reason to, right? But it turns out it is. They are not exactly the same, but so this should be the similar symbol. Somehow the number of A's is very close to the number of T's, and the same is true between G and C. And he cannot explain why, but it's true throughout. So and this particular relationship is called Shargov's second the parity rule. Now we can ask the question in the advanced step. So if you're counting number of A and T's, these are single nucleotides, or it's called the monomers. Monomer is for a single one, right? So if you have A, C, D, G, C, then this is called a monomer. Meaning single. So if you have a one base and base 1 and base 2, two bases pairs. It's called I think it's called the bimer. I'm not quite sure because yeah, I need to double check that one. And if you have a three bases in succession, it's called a trimer. And quad is five, right? Quattromore, I don't know. I don't know what they're called in the 4, 5, and 4, but you follow these. Is this Greek or Latin? Yeah, so Greek counting words for the particular number of the bases in the group. So any one of these is called a oligomer. So when you talk about an oligomer, it can be a monomer, single nucleotide, or trimer, or the group of 4 bases, or 5, and so forth. So it's for a generic term. So for a monomer or single base, you have these two properties by Shargov. How about if you have an oligomer? So for example, trimer, if you have a look at the group of 3 or window of 3 bases, and then start counting them all the way across the double strand or single strand, would there be similar relationship like this? It's a simple counting problem. So if the oligomer length or window size is k, then this k, in this example, k is equal to 3, trimer. Then this slides along the sequence, one base at a time, and count the particular the trimer and see how many of them take place in the given sequence. Then it turns out that it has this particular shape. So these are in different chromosomes in the east, but regardless of which chromosomes you have, they have a fairly identical shape. So in order to get to this particular shape, I think I have that. So tough to do it in the Zoom. A bit more. And also my screen is fractured, so doesn't help either. Blow it up. Okay, so these are the number of counts. So if I take a look at the counts of TTC, then it has about 25 to 25. Yeah, but give it 25. What is the values there? Yeah, anyways, number of counts. So if you scale it, let's say it's at 25, a little bit less than 25 for TTC. And if you go across, then you have similar counts for GAA and CAA, like that, right? So if you take a relationship between the mirror images, the count n is a mirror image, then we have a, let's see, in this example, more, where is it? Oh, where'd it go? Yeah, so you have I picked a TCC, right? So TTC has count of about 25. And if you go across in the mirror image, then you'll find GAA. GAA also has count of about 25. And if you have a count of, I don't know what is this, about 15 in GCA. And if you go across in a mirror image, then you're going to see TGC. In other words, counts on GAA is very similar to count of TTC. GCA is very similar to count of TGC. Can anyone put a comment on the chat or speak out? What are the relationship between these two? This one versus that one, and this one versus that one. So what are the, what is the pattern we can find that these two sequences have approximately equal number of counts as long as two sequences have what kind of relationship? Can anyone see it?
So they are the counterpart of each other, but in opposite sequence.
Yeah, they are complement of each other, but in reverse order, right? So if about GAA, if you write them backwards and then find their pairings, TTC, then there we go, right? So these are the reverse complements. So as long as the oligomers have this reverse complement relationship, their properties turn out to be about the same as far as the count goes. And that is also true for, zoom out a bit, other organisms. So we had, which one was it? The yeast, we don't have that. So the E. coli has this dark green shape. So it's a more evenly spaced. And humans are the light blue. So going, I think this one is human, right? And C. elegans is the crawling worm. So worms have very sharp disparity among the counts, right? And things like that, but they still show this distinct symmetric pattern. Why is it? We don't know, and biologists don't know, but it is there. Okay, so that's what it is. And compared to RNA, again, the T changes to, or thymine changes to uracil in RNA, thymine in DNA changes to uracil. But RNA is single-stranded. Some RNAs are double-stranded, if not entirely, but some portions of it. But the RNA is basically single-stranded, and the DNA, since they are double-stranded and then bonded by the base pairings, they are very stable. Whereas RNA is, since it's single-stranded, is very likely to change because some other atoms or bases can be combined and then changes shape and property right away. The particular data, the RNA type that we're going to run into a little bit are the transfer RNA, and this is to the bind amino acid that we're going to talk about later on during the protein synthesis. And that's why it's called the transfer RNA. So it attaches the particular trimer sequence with the corresponding amino acid according to the genomic code, genetic code that the homolog 1 is about. And ribosomal RNA, and this is used to create the messenger RNA. And messenger RNA is, you heard about the mRNA, right? So the COVID vaccines or mRNA vaccines and so forth. And this is the messenger RNA. So this is the actual RNA sequence that manufactures the proteins corresponding to this particular RNA sequence or gene sequence. So the messenger RNA is a copy of gene sequence, and from this copy of gene sequence, particular gene sequence, the ribosomal RNA. Now so from the DNA sequence, genomic sequence, the ribosomal RNA will pull out, manufacture this messenger RNA, and from messenger RNA, all the relevant amino acids and proteins will be manufactured and created. And those proteins are what actually processes the biological function corresponding to the particular gene sequence. And we'll take a look shortly. So genes to proteins. Okay, so are there any questions so far? So are you all good? Genes to protein. So this is the most critical thing, right? So we want to find out how a particular biological function works from a gene. Right? So when we eat some food and it has to be digested, then all the proteins have to be working together to digest your food and then separate out the nutrients and throw out the rest and things of that nature. So how does all those proteins are generated? That's what you're going to do. So this is the central theorem and central dogma of the molecular biology, and it goes through the basically two steps. So from the DNA, where the genome is, and DNA is nothing but a static map of what kind of genes you have in your DNA or in the chromosome or genomic sequence. So from the static DNA sequence, if you have a cell that performs a particular function, then you need to make or manufacture the proteins to perform particular function, and that is going to be copied from the map into dynamic map of RNA. Okay. So the gene is copied out into RNA, or it's called expressed or activated. And then from the RNA, a number of different proteins will be manufactured or generated. So the group of these proteins are the ones which actually perform the associated biological function from the RNA or that particular gene. So the copying of gene sequence into messenger RNA is called transcription. So you're transcribing, making a copy from static gene sequence And once we have a messenger RNA, then you're going to manufacture a number of different proteins. Okay, so you can take a look at those animations. I think they should be all available. I didn't check them, but should be all set. So from inside a cell, nucleus, you have a chromosome or DNA. And from here, a particular gene sequence will copied out, and it will be pushed out of the cell, the nucleus. And here, a number of different proteins will be generated. Okay. So for these two steps to be done, the first order of business is that remember that DNA is double-stranded and they are wound up very tightly, right? So the first thing you have to do is break up the base pairing between two strands, and then make a copy from the gene and pass it out as a messenger RNA out of the nucleus boundary to the messenger RNA. And translation is to generate the sequence of amino acids, which is called the proteins, from this messenger RNA sequence. So the first thing we're going to ask is the following. So we have a double strand. Now they are not really the so you have a double strand. And for simplicity, suppose the gene you want to make a copy of is ATCG. Make it very simple. Okay, so this is the gene sequence, and obviously it'll be much longer than this. So we locate a gene in this particular strand, and we're gonna make a copy of that and pass it out of the cell, the nucleus boundary, as a messenger RNA. Okay. How are we gonna make a copy of it? And we know the messenger RNA will have a ATCG. Turns out that what they do is, if this is the gene that we want a copy, then in the other strand, so if we call this Watson strand, Crick strand is the opposite strand. So in the Crick strand, what do we have? We know that it has the complement of this particular gene sequence, right? So what it can do is they are initially bonded together, but after we break that up, if we can generate the complementary sequence from this opposite strand, then we have the same sequence as the gene sequence. So that's the ATGC. That's what it can collect and generate out as a messenger RNA. So if you want to have a copy of a gene from Watson strand, then we actually make a copy from the Crick strand, and then Crick strand into messenger RNA. So fairly ingenious way of doing it. So this one is show you. So if you have a gene sequence in the non-coding strand, then the opposite strand we generate or make an extra base pairings from the upper strand, and collect them into messenger RNA. So the same idea there. But once you have a messenger RNA, which is copy of gene sequence, then how do you create proteins or amino acid? And it turns out there is a particular coding map between the nucleotide bases to amino acid. Yeah. And we have only 20 amino acids in the world. There are probably not. In the biological world, there is 20 amino acids. And 3 bases are combined to produce single amino acid. So this is the sort of the table. So if you have the base sequence of U, the same as T in DNA, U, then the amino acid, I didn't bring the sheet. I don't know the name of PHE is generated. And if you have a UUA, then leucine amino acid is matched up, and all leucines, and valine GUU is valine and alanine and the and so forth. Okay. So if I have 3 bases, bases base 1, 2, 3. And each base is one of the 4, right? 4 nucleotides. So we have a 4 times 4 times 4, this table, which is 64 different combinations. And this trimer, or 3 bases, called the codon. So from one codon, so we have 64 different codons, but we only have 20 amino acids. So this is the multiple to 1 mapping, not a 1-to-1 mapping. So as you can see, these 2 are mapped to PHE, the protein. You have 6 of them, regardless of these particular codon sequences, they all produce leucine amino acid. And all of these produce proline amino acid, glycine and arginine. Arginine, actually 6 of them. And it's, yeah. So this is sort of a degenerate mapping, degenerate coding mapping. So from 64 possible triple codons, you only have 20 amino acids that they produced. So the the codon to amino acid mapping, this table is called a genetic code. So it may not be a good terminology, but it's called a genetic code. And there are a number of studies for a long period of time how this genetic code came about and what the genetic code might have been like in earlier ancient times when there is a very limited species and things of that nature. But again, it's not it. So from codon, triply called codon, 1 amino acid is attached, sort of, connected. And that process is done by ribosome and also transfer RNA. We'll see that in a minute. Okay. Okay, I think we can take a look at this YouTube. Or
Fred Smith here told us the B&H photo helped change his life. Fred, tell us your story.
Hello guys, in this video we're going to talk about synthetase, the enzyme that links amino acid to tRNA. We're going to talk about its structure, classes, and role. When tRNA binds with its anticodon to the mRNA codon, it brings with it the corresponding amino acid attached to the 3' OH of tRNA terminal adenosine. But how does the amino acid get attached to the tRNA? Well, it's done with the help of an enzyme called amino-acyl tRNA synthetase. This enzyme catalyzes a two-step reaction. First, the carboxyl function of the amino acid must be activated by adenylation. Then, the activated amino acid.
this is the transfer RNA structure. And what she's trying to say is basically, this is so dark here. At the bottom here is the I don't know whether see any picture. Yeah. So at the bottom of the transfer RNA, you have what's called an anticodon. Anticodon is a reverse complement of the codon. Okay. So the codon sequence will be attached to this anticodon sequence because they are complemented with each other. And then depending on what codon you have or anticodon you have, it'll bring in or attach a particular amino acid. So from the codon to amino acid, there is a particular mapping, and that is the genetic code that we have. So you can take a look at this video later on. So the anticodon is reverse complement of codon, and this part of the transfer RNA. And depending on what you have in the anticodon, that transfer RNA brings or attaches specific amino acid. Namely, depending on what codon is, it attaches a specific amino acid. Right? So that's the translation process. It's the same table in a different form. And when you have a messenger RNA, depending on where you start, because you can have a one codon here, or the first one may not be just the part of the encoding process, and codon may start from the next, the nucleotide, and then forth. So you have three basic reading frames where the actual amino acid will be starting off. So even though you have one sequence, you need to find out which particular if the nucleotide is the beginning, the codon sequence. Amino acid will spend some time later on. Okay. So this is the common amino acid structure, and depending on what is attached here, you have 20 different amino acids. So residue what's attached, what consists of, what constitutes this residue R will basically determine what amino acid you have. But you always have this the carbon, alpha carbon at the sort of in the center, and then you have hydrogen and nitrogen and so we'll come back to amino acid later on. And if you look at the entire genome sequence, genome size, okay, E. coli is a bacteria, basically. Bacteria is about 4-5 megabase pairs, megabases, million base pairs. Yeast is the smallest living organism. Has the biological name of this one, right? About 12 megabase pairs. And nematode or C. elegans is again the crawling worm. So 100 megabase pairs. Thylia, the Thylia now is the flowering plant, which has been studied extensively. So this is the plant basis. But 115 fruit fly. Yeah. The humans now jump to 3 billion. Okay. So most the simpler organisms are in the yeast is really tiny, but about 100 megabases and so forth. But if you look at the number of genes contained in each genomic sequence, virus about 80, bacteria the 4,000, and so forth. So the genome size jumps to 3 billion for human, and since they have 20,000 or so genes, people initially thought that the human should have about at least 100,000 genes contained in that sequence, because you have an order of magnitude increase in genomic sequence, and you should have about same ratio for the number of genes. But it turns out that humans have only 20,000 over, I think that now it's about 22,000 or so genes in the sequence. And clearly that is not logical, because if you compare the different levels of intelligence and functionalities and so forth, the natural question is, how can humans do all the things they do with the approximately same number of genes that in these lower organisms are doing? Okay. And this is just a sort of an outlier. The fugu, the fish, has actually has more genes than humans, and wheat has a humongous the genomic size. So you don't see that many wheat sequences, gene sequences constructed. The rice also has a much longer genome size than humans. And they have more genes than humans. So it defies the logic somewhat. So with the small number of genes, particularly the humans can have this multiplicity of functions, and that has to do with what's called the splicing, alternative splicing. So when you have a gene sequence, you don't produce one protein from one gene sequence. You actually have a combination of small subsequences combined to produce one protein, and different combinations produce different proteins and so forth. And this process is called alternative splicing. And what are combined into proteins are called exons. So exons in pink. And between exons, you have what's called intron sequences. So introns are not involved in protein sequences. They are kind of boundaries between successive exons. So in a given gene, not the entire gene sequence is coded or involved in protein manufacturing. Somewhat allergic. and in particular, the first part, which is called UTR, untranslated region. So this 5' is the front end, and 3' is the trailing end, right? So they are separated by these two UTRs, and within the coding region, again, the exons are combined into different proteins. Yeah. And that's how, with a small number of 20,000 genes, humans can have such a wide variety of functions. Same idea here. This is, again, the same idea, capping. So from the DNA gene sequence, which has the exons separated by introns, they're first transcribed out into primary mRNA, and then ends are kept, and this will be the messenger RNA, and they are spliced into different proteins. Right. So the same thing in different picture. So the transcription is done again by copying from the opposite strand of the gene where the gene is contained. And as you do, you need to first split the double strands. The splitting is done by the RNA polymerase. So it cuts it open. And as it cuts it open, this ribosome will travel to produce copy RNA. I'm going to wait to that. So, sorry, the RNA polymerase. Yeah. Okay. So the RNA polymerase, there are the couple of compounds in the RNA polymerase. It first cuts it open, and then it does the base pairing, and produces the RNA. And ribosome is actually used in the protein synthesis. By putting messenger RNA and transfer RNAs and all those in the right positions, and start producing the amino acids. Okay. So again, to recap, sort of, messenger RNA is produced and transported out of the cell nucleus. And that's where the messenger RNA is translated or alternatively spliced into multiple proteins. And as you do the translation, transfer RNA is involved. Again, you need to look at the trimer, the bases, and then attach that and find out which amino acid is called in. And I think I had a sorry, I didn't double check the animation YouTube. Yeah, here it is, right? I thought there is a better animation than this one. I'll find out. Yeah. So yeah, there is a animation for that. So transfer RNAs is a very, very small RNA, but it plays a critical role in converting RNA sequence into amino acid, corresponding amino acid. Again, then mapping is done between codons to anticodon to particular shape of this amino acid, the transfer RNA. So we could go over those U2 and as forth, but I'm skipping over those. So we have this article, which will be your homework 1, written by Douglas Hofstadter in early 90s. Yeah, that's correct. I think it's later, even earlier than that, early 80s. And somehow I kept a copy of that. So I looked at some of the interesting articles, and he's a logician. He's a mathematician in logic. So his argument is very interesting. And let me take a look at this one. What does it have? Whether this has a more exciting animation or not. So let's try that. No. So it's a transfer RNA sequences. It's not that interesting. Okay. So he poses an interesting question of how that particular genetic code table came about, or did it come about by the design of a god, or some higher being, or this somehow came about on a random basis or arbitrary. This obviously must have some chemical reasons, right? Because these are nothing but the chemistry. But how this particular map or table came about is still a mystery, and what people are still studying, the origin of this genetic code and so forth. And he poses a very interesting logical question. And that is, this is the known working genetic code for the organisms, but can we use any arbitrary genetic code table to replace it and still come up with the same mapping? In other words, what constitutes, or what are the key elements or key components of converting from this the codon sequence to amino acid? And that's where the again, that transfer RNA is involved. And I think yeah, this one is a better diagram. So from the codon sequence to amino acid, and what makes up the matching between codon to amino acid. And what I was, it turns out that there is this DH loop involved and so forth, and also enzyme called amino acid tRNA synthetase, which recognize this particular DH U loop, okay, D loop, what they call it D loop. And he makes an interesting argument. So the homework 1 is to read Hofstadter's article and find out the first summarize what the role of amino acid tRNA synthetase is. Okay. And then the restate was his hypothesis and how he proves or disproves his hypothesis. And if you agree with this claim, then state why you agree with him. Otherwise, what your argument is. Right? So that's the homework 1. Right. again, so if if you had in the classroom, then yeah, I could use the board screen and so forth and play those YouTubes. But I think that today is a snowy day, so I want to make it short and stop here for the first class. Okay, any question? In the Blackboard, you should have an assignment 1, homework 1, actually. And you can submit your PDF or Microsoft Word in the homework 1. Any question today?
Professor, how can you tell which is the coding strand and which is non-coding strand?
Let's see. Okay, let me get back to the desktop. Okay. And it depends on where your gene is located. So I think that if you have a Western strand, then the direction will be, well, they have different directions. Direction goes that way, and the opposite strand will go in that way. Okay? So if your particular gene sequence is ATCG, for example, then this is the gene you want to make a copy of. Then you go to the opposite strand, and then you start. I think they do different way, do in chops, but basically they create a copy of your complementary strand to have an exact copy of a gene sequence, and then you take that out as a messenger RNA, mRNA. So which one is the coding strand, non-coding strand? It depends on where gene is. So if the gene is located in this strand, this will be non-coding, because coding of gene is not done from this strand, but from the opposite strand. So this will be a coding strand. Gene is located in the Crick strand, then this will be non-coding strand.
But how do you know where the gene is located?
Oh, you have the entire genomic sequence or the or decoded, right? So actually, we'll take a look at it when you actually do the BLAST search and so forth. Then yeah, we can see where the gene is. And they are all annotated. So given the chromosome 1, right? Chromosome 1, certain the base position to which other positions we have, then they are all annotated now, which genes are there and what the role of that gene in with respect to what proteins is involved in producing and so forth. Actually, we can go to West. To, I don't know whether it's the right one or not. There's some searching part, right? Let's see. Maybe I could go to particular protein. Yeah. The name of that database. Okay. Forgetting a lot of things nowadays. Try to look at the protein database. Let me take a quick look at. I have CNN data. See. Just forgot where that protein database is. Let me take a quick look at. Not it. That's what I'm. What I can do is take a look at the other size. Add. Should get proteins, right? Is it? Yeah, it's for an alignment. Would that be? Protein database. Start with the Pro. Got it. Sequencing. Got it. Yeah, I'm sure it'll come up. So I'll start from there next week. Okay. Start with the protein database, and given the protein name, it gives all the sequence information. So I'll pick up from there next week. Any other question? No? Okay. So take a look at the article by Hofstadter, and yeah, that is a very interesting article, and that will give you a very good idea about the translation, transcription, and translation, the gene code to protein manufacturing process. Okay, so see you guys next week in class, and yeah, so.
Thank you, Professor.
I'll see your the homeworks. Okay. Okay. Thank you.